Итак, уважаемые коллеги, мы просим всех занимать свои места в зале. Рабочая сессия начнется ровно через 2 минуты.
Доброе утро. So, good morning, ladies and gentlemen. So, for who wants some kind of headphones for interpretation, so we will bring some for you just in a couple of minutes. And I will start speaking in Russian in a few seconds, and we'll introduce first speakers and some of our guests to give a kind of an introduction and greetings for the audience. Так, уважаемые коллеги, сейчас раздадут наушники для перевода для наших иностранных участников, и мы начинаем. И первого на сцену я хотел бы пригласить для открытия конференции ФизтехБио первого проректора по научной работе МФТИ Олега Анатольевича Горшкова. Поприветствуем.
Дорогие гости, уважаемые коллеги, я рад приветствовать вас сегодня в этом зале по случаю открытия конференции ФизтехБио, посвященной работам в области живых систем. Это уже 4-я конференция, и можно утверждать, что сложился свой формат конференции, когда обсуждаются не только результаты современных исследований в области, в частности, биотехнологии, биоинформатики, медицинской техники, но также обсуждаются вопросы практической реализации тех результатов исследований, которые достигаются в научных лабораториях, и это всегда важно, я имею в виду практическое внедрение тех результатов, которые получаются в науке. А в данном случае, когда речь идет о таких направлениях, как фармацевтика, медицинская техника, это, наверное, вдвойне важно, потому что вопрос касается обеспечения здоровья людей. Надо сказать, что у нас на физтехе этому направлению живых систем в последнее время уделяется большое внимание. В прошлом году, в частности, у нас создан специальный факультет биомедицинской физики, который призван объединить все наши усилия в этом направлении, по крайней мере, в части обучения. В настоящее время при поддержке Министерства промышленности и торговли ведется строительство здания для биофармкластера «Северный». У нас создан в Институте Центр живых систем, призванный также объединить наши усилия в области исследований в этом направлении и практического применения результатов. И ряд других направлений. Кроме этого, у нас в МФТИ в настоящее время реализуется программа повышения конкурентоспособности университета. Это делается при активной поддержке Министерства образования и науки. В рамках этой программы заметное внимание уделяется направлению живых систем. Ну и в частности, вот эта наша конференция проводится при некоторой поддержке со стороны этой программы. Есть существенная часть программы, связанная с созданием новых лабораторий здесь, на физтехе, и при этом снова заметное внимание уделяется физике живых систем, создаются лаборатории, не только научные, но и те, которые ближе к применению результатов исследований на практике. Поэтому можно говорить, что в настоящее время это направление физики живых систем является важным и существенным не только в других организациях, но и на физтехе, где нельзя сказать, что это направление является традиционным, тоже является в настоящее время важным. Уважаемые коллеги, я хотел бы пожелать вам плодотворной работы, творческих дискуссий, появления новых идей, личного общения в перерывах и вечером, то, что является всегда неотъемлемой частью всех конференций. Я надеюсь, что время, проведенное здесь, для вас будет полезным, и следующая конференция, которую мы обязательно будем организовывать, будет также интересной для вас. Вы найдете время приехать к нам, рассказать о своих результатах, и вместе мы сможем усилить это направление не только здесь, на физтехе, но и в стране. Спасибо за внимание.
Спасибо, Олег Анатольевич. И на сцену я хотел бы пригласить Олега Ивановича, главу города Долгопрудного, для приветствия участников конференции.
Добрый день, дорогие друзья, уважаемые гости. Мне очень приятно сегодня в рамках руководства города, на котором сегодня, на площадке которого проходит и будет проходить вот эта конференция, поприветствовать вас всех. сказать особые слова благодарности руководству института за то, что вот это происходит у нас, это движение, этот рывок вот в самом главном, в стремлении оздоровить и сделать здоровым наше человечество. Это очень важное направление. Ну и из истории, если говорить о том, что обычно прорывы в науке происходят на стыке веков. Вот мне кажется, сегодняшний день это как раз вот тот шаг, в котором молодежь, вот те молодые сотрудники, ученые люди, которые находятся здесь, у нас в городе, на этой площадке, смогут это сделать. Для города это очень интересная тема, это очень интересное направление и развитие. Мы сегодня вместе с институтом, вот с Иващенко Андреем Александровичем делаем в этом направлении определенные шаги для того, чтобы, кстати, шаги для того, чтобы заложить основы этого прорыва, потому что буквально в этом году будет открыта здесь школа для высокоодаренных детей. Детей не только нашего города, вот та система многолетняя, больше 10 лет, которая работала и работает сегодня у нас в школе ФизтехШкола, она уже будет работать школа ФизтехШкола области, областная школа. Вот сюда будут прибывать дети из Московской области. И мы, и правительство Московской области, и наш губернатор Андрей Юрьевич поддерживает вот эти начинания, все вот эти стартапы, которыми сегодня вы занимаетесь. Я считаю, что для нашего города сегодня и вообще развитие науки, развитие физтеха, это как, вы знаете, можно сказать, что умный Долгопрудный, умный физтех, умная социальная среда. Но самое главное, что я считаю, что это умные люди, умные дети, те, которые учатся сегодня в стенах этого института и которые будут двигать эту науку и создавать эти прорывы. Потому что и отсюда, из этих стен, есть нобелевские лауреаты. Я сегодня знаю, что здесь тоже будут в работе конференции принимать эти замечательные ученые. Я считаю, что это очень важно в стенах этого института, в стенах нашей Подмосковья, в стенах Российской Федерации. Желаю всем хорошей работы. Я надеюсь, что у всех, особенно у гостей, останется хорошее впечатление об этой конференции и о том, что здесь будет совершено. Спасибо, успехов.
Уважаемые коллеги, прежде чем мы начнем, небольшая информационная справка про нашу конференцию. Во-первых, в нашей конференции принимают участие два нобелевских лауреата. Это Роберт Хубер и Майкл Левит. У нас есть иностранные ученые из США, Норвегии, Германии, Бразилии, представители фармацевтических кластеров России, научно-исследовательских институтов, университетов, компаний-разработчиков. На конференцию зарегистрировалось порядка 500 человек. Я думаю, несмотря на дождь, будут подъезжать новые и новые люди. И я хотел также выразить благодарность нашим спонсорам. Это компания «Эбот», золотой спонсор конференции, это компания исследовательский институт химического разнообразия и фарма, и компания EMC, которые стали серебряным спонсором нашей конференции, и генеральный информационный партнер Интерфакс, а также научное кафе Science Action. Соответственно, я на сцену приглашаю первого спикера, это президент правления Центра живых систем МФТИ Андрей Александрович Иващенко. Он сделает небольшой доклад про Центр живых систем МФТИ, о целях и задачах, которые стоят перед центром, о том, какие лаборатории уже созданы, что создано в этом направлении и какие планы нас ожидают в ближайшее время. Андрей Александрович Иващенко.
Уважаемые коллеги, уважаемые гости конференции, спасибо огромное за то, что вы в этот дождливый день нашли время доехать в Долгопрудный, куда, как поется в известной песне, город чудный, и не так просто добраться. Но вот благодаря тому движению, которое происходит вокруг физтеха, ядром которого выступает наш любимый Московский физико-технический институт, потихонечку этот город начинает превращаться в то, что мы надеемся станет современным технополисом, вокруг университетским технополисом. И мы все будем гордиться и уже гордимся всей этой местностью, потому что это действительно будет такой прорывной проект российской модернизации. Я расскажу, поскольку наша конференция посвящена живым системам, биологии, я коротко постараюсь рассказать, чтобы не отнимать время от докладов наших уважаемых гостей, нобелевских лауреатов, о том, что уже делается. Вы, наверное, кто-то, кто здесь уже был, видели, что это все происходит буквально последние 2 года на физтехе. И это действительно стартап. Еще очень много что не получается, очень много сбоев. Но вот та энергия, которая здесь есть, и та скорость, с которой происходят изменения, конечно же, очень внушает оптимизм многим, и мы верим, что у нас все получится. Я хотел бы... Как переключать?
Нет, говорите следующий слайд.
Тогда я буду говорить следующий слайд. Несколько слов. У меня слайды на английском языке, поскольку многие из наших гостей англоговорящие, но я буду говорить по-русски, и я надеюсь, что переводчики правильно все переведут. Несколько слов про МФТИ для тех, кто первый раз здесь и не знаком с нашим институтом. МФТИ был организован после Второй мировой войны в 1951 году. Вначале это был факультет МГУ, а потом он выделился в отдельный физико-технический институт. Система физтеха известная тем, что это всегда была сетевая организация, которая отбирала самых талантливых детей, и благодаря руководству института до сих пор сюда идут ребята с самым большим ЕГЭ в стране по физике, математике. Ребят первые 3 года учат фундаментальные науки, они осваивают программу физфака и мехмата, сжатую в 3-летний курс, то есть нагрузка здесь по сравнению с Московским государственным институтом раза в 2 больше. И дальше после такого фильтра на трудолюбие ребята встраиваются в различные базовые организации. Во время Советского Союза это были организации индустриальные, промышленные, Академия наук. Конечно, вместе с тем, как изменилась наша страна за последние 20 лет, часть этих организаций исчезла, но появляются новые организации, новые партнеры, которым нужны вот такие талантливые выпускники, которые начинают встраивать их в свои системы, и, соответственно, они уже, работая там, осуществляют научные исследования, внедренческую деятельность и модернизацию нашей экономики. Поскольку физтех сейчас участвует в программе по повышению конкурентоспособности, совсем недавно был сформирован международный совет, научный совет, который возглавил президент MIT доктор Рафаэль Райф. И туда вошли очень выдающиеся ученые, в том числе и Нобелевский лауреат, как российские, так и иностранные. Следующий слайд. Физтех, конечно, всегда в основном готовил ребят с территории бывшего Советского Союза, России, но потихоньку здесь появляются и студенты из различных других стран, то есть растет международная мобильность и международная составляющая кооперации, как с точки зрения студентов, так и с точки зрения профессоров, которые привлекаются для преподавания или, наоборот, направляются преподавать в другие университеты. Следующий слайд. Традиционно система физтеха строилась на том, что основатели ее были нобелевские лауреаты. Вот вы здесь видите, это те люди, которые за историю физтеха преподавали или формировали курсы, имели отношение к формированию физтеха. И это очень важно, что ребята на старших курсах могут работать с практическими учеными и вот в режиме мастер-подмастерья перенимать вот эту культуру работы в науке, во внедрении. Также выпускники физтеха, как вы знаете, в 2010 году вот профессора Гейм и Новоселов получили Нобелевскую премию за графен. Это, конечно, предмет нашей особой гордости, и мы понимаем, что своего рода это некий такой своеобразный знак качества той системе образования, которая здесь сложилась, потому что даже сейчас она, несмотря на все те переломы, через которые прошла наша система образования в последние годы, продолжает работать на очень высоком уровне и готовить хороших специалистов. Следующий слайд. Теперь переходим к живым системам и к тому, как меняется физтех вот в условиях новой рыночной экономики. Раньше на физтехе практически не было науки. Вся наука была в базовых кафедрах, в базовых организациях. Сейчас в дополнение к тем базовым кафедрам, базовым институтам, которые остались и успешно функционируют, начинают появляться исследовательские лаборатории и исследовательские центры непосредственно в кампусе МФТИ. И это очень важно, потому что те люди, которые занимаются практической наукой, они начинают больше общаться со студентами, больше преподавать, вести семинары, практикумы, идет такое живое обучение, живой науки. Вот если говорить о живых системах, вот здесь вот на карте приведены основные объекты, которые появились за последнее время. Это и новые лаборатории нашего исторического лабораторного корпуса, это и биобизнес-инкубатор, который в корпусе прикладной математики был 2 года назад открыт с участием Нобелевского лауреата профессора из Крипса Барри Шарплеса. Это геномный центр и центр клеточных технологий, который открылся только в конце прошлого года. И, конечно, самая главная инфраструктура – это вот корпус, биофармкорпус, который, если вы выходили и выйдете из этого здания, посмотрите налево, он достраивается и будет запущен с начала следующего года. Это по программе при поддержке Министерства промышленности и торговли. Осуществляется это строительство. Это будет такой настоящий исследовательский институт, где будут потом сконцентрированы все эти лаборатории и будут вестись исследования мирового уровня. Уже сейчас у нас работают, несколько слов о том, какие уже сейчас есть лаборатории. Сейчас уже работает лаборатория по идентификации новых мишеней, биомишеней, в том числе GPCR-белков. Следующий слайд. Активно развивается направление медицинской химии. Следующий слайд. Очень серьезное аналитическое оборудование поддерживает как химическую аналитику, так и биоаналитику, клинические испытания. Есть лаборатория высокопроизводительного скрининга. Со всеми этими лабораториями за время работы конференции, я думаю, будет возможность ознакомиться и поговорить с руководителями, которые работают. Одна из новых лабораторий – это longevity стадии продуктивного долголетия. Как меня вчера правильно поправили, нужно не продуктивное, а репродуктивное долголетие для страны, поскольку оно гораздо более важно. Я тоже с этим полностью согласен. Есть лаборатория готовых лекарственных форм, которая активно сотрудничает с российскими и зарубежными фармпроизводителями, помогаем формировать новые портфели фармпрепаратов. Геномный центр только открылся, и целый ряд проектов инициировано на новейшем оборудовании. Следующий слайд. Центр клеточных технологий вот открывается сейчас буквально. Следующий слайд. Мы также активно работаем с производителями медицинской техники, медицинских приборов, и они тоже открывают, являются членами биофармкластера и начинают открывать свои лаборатории, делать инновационную продукцию, используя вот тот потенциал и ту инфраструктуру, которая здесь создается. Следующий слайд. Ну, это вот этот корпус, которого мы все с нетерпением ждем, 11 тысяч квадратных метров. Думаю, там будет работать около 500 ученых через 2-3 года. На этом слайде перечислены основные наши партнеры, я не буду их всех зачитывать. Основная идея, что Центр живых систем, вот, который был создан на физтехе, он является интегратором разнообразия этих партнеров. Потому что, с одной стороны, у нас есть индустриальные партнеры, фармпроизводители, у них одна повестка дня, одни потребности. С другой стороны, у нас есть партнеры академические, академическая наука, у них свои форматы работы. И, конечно же, мы активно работаем с медицинскими центрами, с врачами, с медиками, у них тоже своя повестка дня. И вот, конечно, это нетривиальная задача объединить вот эти все вещи для того, чтобы возникло реальное разделение труда и синергетический эффект. Вот если Центру живых систем получится это сделать вот в этом месте, в этой точке бифуркации, которая называется физтех, то мы реально увидим прорывной проект мирового уровня. Мы, конечно же, очень благодарны руководству страны. И вот Олег, я думаю, зачитает. Только что поступило приветствие от вице-премьера Аркадия Дворковича в адрес нашей конференции. Он собирался приехать, но, к сожалению, Долгопрудный — это не так близко к Кремлю, а может и к счастью. Вот. Но он действительно энтузиаст того, что здесь происходит, нас активно поддерживает, потому что в рамках его компетенции находится не только развитие фармацевтики, но и всех биотехнологий. Потому что вот то, что мы сейчас видим, начав с применения технологий в области фармацевтики, те же самые технологии начинают быть востребованы, у нас появляются партнеры, которым нужна та же химия, тот же скрининг, те же способы аналитики, но в животноводстве, в сельском хозяйстве, в аквакультуре. Более того, появляются партнеры, которым надо искать новые катализаторы в добывающих отраслях промышленности. Оказывается, что вот эти технологические исследовательские платформы, они вот имеют такое диверсифицированное применение в очень разных областях. Но возникают, конечно, драйверы, это фармацевтика и медицинские приборы. Следующий слайд. Следующий слайд. Несколько слов о том, какие программы уже здесь выполняются. Вот на этом слайде приведено несколько лабораторий, которые поддержаны Министерством промышленности и торговли. Здесь непосредственно в физтехе вы видите 2 фармацевтических проекта, 4 диагностических medical device проекта. Уже получает финансовую поддержку как промышленность, так и Министерство промышленности и торговли и реализуется в наших лабораториях. Следующий слайд. Московский физико-технический институт стал организацией, которая осуществляет по заказу Минпромторга сейчас мониторинг и экспертную поддержку программы «Фарма-2020», что, конечно, с одной стороны является определенным вызовом, потому что, несмотря на то, что у нас много базовых институтов, базовых кафедр, экспертов, но это очень серьезная работа, и она очень ответственная. Мы надеемся, что Министерство промышленности и торговли будет довольно той работой, которую выполняет вот наше экспертное сообщество. И в прошлом году, как вот говорил Олег Анатольевич, команда физтеха под его координацией непосредственной выиграла в конкурсе 5 топ-100, что означает, что сюда стали поступать целевым образом средства из Министерства промышленности, Министерства образования и науки, и в ближайшее время будет создано более 50 новых лабораторий по основным направлениям. Это будут исследовательские лаборатории и лаборатории индустриальные, вместе с индустриальными партнерами. Вот здесь примерно показана скорость создания лаборатории, но уже сейчас ясно, вот тут написано New Labs, что к 2017 году. Я думаю, к 2017 году реально, если будет упущен этот корпус, только, наверное, в одних только живых системах будет столько New Labs. А по другим направлениям IT-телеком, новые материалы, новая энергетика, тоже будут лаборатории. И, конечно, скорее всего, физтех эти индикаторы даже опередит. Следующий слайд. Ну, несколько слов про биофармкластер. То есть биофармкластер был создан в 2010 году. Это некоммерческое партнерство МФТИ и различных индустриальных и бизнес-партнеров, которым интересно на базе МФТИ развивать новые продукты, готовить себе сотрудников, развивать свои компетенции. И поэтому основная миссия биофармкластера — быть вот таким интерфейсом между наукой и индустрией. Следующий слайд. Программа биофармкластеров была инициирована Министерством промышленности и торговли. Вот сегодня у нас присутствуют здесь представители еще нескольких кластеров. У нас впереди это и Санкт-Петербург, и Калуга, и Ярославль, у нас и Казань. У нас впереди предстоит интересные сессии, тоже посвященные обмену опыта в этой области. Следующий слайд. Здесь, конечно, уникальное место. С одной стороны, сюда трудно доехать, с другой стороны, вот сейчас, когда будут достроены эстакады, новая Ленинградка, то место очень удобное, потому что оно по классике, как все технополисы, находится между международным аэропортом и крупным мегаполисом. Плюс здесь традиционно очень высоко образованное население, потому что еще в советское время тут находилось огромное количество различных НИИ, исследовательских центров, поэтому как бы человеческий ресурс, человеческий капитал, как это называется, здесь находится. И, конечно, самое важное — это наличие такого ведущего университета, как источник живой воды, как такой ключ, который бьет молодыми талантами, и все вокруг хорошает. И мы надеемся, что все здесь будет цвести вокруг этого источника живого. А это просто здесь приведены направления, в которых еще создаются кластеры. Везде есть индустриальные партнеры, крупные, известные, менее известные. И поэтому вся эта программа, она осуществляется в рамках общей программы создания научно-образовательного кластера «ФизтехБио» в московском регионе вокруг Московского физико-технического института. Следующий слайд. Ну, я не буду останавливаться, здесь слайды как бы работаем с различными партнерами. Наверное, мы их давайте пролистаем, чтобы не тратить время. Это кому интересно, мы можем поделиться своим опытом. У нас есть опыт работы и с Big Pharma, и с американскими университетами, и с российскими организациями. И, в общем-то, сейчас уже понятно, что получается, что нет, что можно масштабировать, а что, как бы, может, было ошибочно. Но, тем не менее, как говорится, любой опыт, он полезен для развития. Спасибо большое. Я постарался как можно быстрее закончить свое выступление. Извините, если затянул.
Да, как сказал Андрей Александрович, поступило приветственное слово от Аркадия Владимировича Дворковича, заместителя председателя правительства Российской Федерации. Я его зачитаю, с вашего разрешения. Уважаемые участники конференции ФизтехБио, перед Российской Федерацией стоят амбициозные задачи по модернизации ее экономики и созданию новых высокотехнологичных рабочих мест. Фарм- и медпромышленность – это одно из приоритетных направлений развития российской экономики, чей инновационный потенциал напрямую зависит от количества новых продуктов в портфелях российских компаний. Разработка инновационных лекарственных средств и медицинских изделий – длительный, высокорискованный, капиталоемкий процесс, который требует слаженной работы всех его участников. Именно на примере этой отрасли нам предстоит модернизировать и построить инфраструктуру мирового уровня, от разработки новых образовательных программ и создания научно-исследовательских центров до строительства новых высокотехнологичных производств и современных экспериментальных медицинских центров. От имени правительства Российской Федерации, от себя лично, приветствую вас на конференции и желаю продуктивной работы и приятных впечатлений от новых встреч. Аркадий Владимирович Дворкович. Уважаемые коллеги, у нас есть еще приветствие от Министерства промышленности и торговли. Я попрошу Алексея Викторовича Алехина, начальника отдела фармпромышленности, зачитать его.
Добрый день, уважаемые коллеги, уважаемые участники конференции. От имени Министерства промышленности и торговли Российской Федерации позвольте поприветствовать вас на открытии международной конференции при Московском физико-техническом институте, который является очень важным партнером Министерства промышленности и торговли. Причем партнерство это осуществляется не только в рамках строительства Центра живых систем, не только проявляется в поддержке различных инновационных проектов, но хочу отметить, что между Министерством и МФТИ подписано соглашение, которое определило основные направления развития системы подготовки высококвалифицированных специалистов в области государственного управления в промышленности. Говоря о проектах в МФТИ, на сегодняшний день реализуется 7 R&D проектов в таких областях, как онкология, заболевания системы кровообращения, пульмонология и так далее. И именно эти терапевтические области являются важнейшим приоритетом стратегии здравоохранения Российской Федерации. Это не случайно. Вся стратегия развития фармацевтической и медицинской промышленности построена на основных приоритетах здравоохранения. И важно отметить, что, вот как Андрей Александрович сказал, что множество проектов реализуется в тесном взаимодействии с врачебным сообществом, в ходе которого возникают многие оригинальные, не имеющие мировых аналогов решения. Нельзя недооценивать опыт наших иностранных коллег, которые обладают уникальными знаниями и компетенциями и готовы ими делиться как здесь на конференции, так и, возможно, в дальнейшем в ходе реализации проектов по техтрансферу. Реализация таких проектов стала возможным в том числе благодаря мерам поддержки в рамках федеральной целевой программы «Фарма-2020», а также с помощью привлечения других финансовых инструментов, какие есть у Сколково, РВК, Роснано, Веба, то есть институтов развития. В последние несколько лет государство активно поддерживает формирование инновационно-внедренческой инфраструктуры параллельно с процессом модернизации предприятий. Это привлекает и частные инвестиции в инновационные территориальные кластеры, где государство в том числе финансирует создание центров на базе ведущих научных и образовательных учреждений. И стоит отметить, что в ближайшие 2 года такие учреждения откроются как вот здесь, да, в МФТИ, это Центр живых систем, так и на базе МГУ, Уральского, Казанского, Ярославского государственных университетов, а также Санкт-Петербургской химико-фармацевтической академии. Знаю, что сегодня коллеги выступят с докладами по теме фармацевтических кластеров в отдельной теме, в отдельной сессии. К слову, ведется активная работа и на базе биофармкластера «Северный», где в рамках федеральной целевой программы также реализуется немало инновационных проектов с участием резидентов. Мы, как Министерство промышленности и торговли, заинтересованы в первую очередь, чтобы новые разработки, инновации, зародившиеся в том числе и на базе научных учреждений, доходили до рынка. Напомню вам, что совсем недавно, 16 мая этого года, в Санкт-Петербурге прошло заседание Президиума Совета при президенте Российской Федерации по вопросам инновационного развития в области фармацевтики и медицинской техники. На нем председательствовал Дмитрий Анатольевич Медведев. И по итогам этого совещания, на котором обсуждались вопросы доступа на рынок, министерству было поручено разработать ряд нормативно-правовых актов, стимулирующих развитие инноваций в России. Это и преференции при государственных закупках, дифференцированные в зависимости от глубины технологического процесса, это и долгосрочные государственные контракты. Также обсуждались вопросы введения преференциального налогового режима для компаний, активно инвестирующих в R&D, и развитие экспортного потенциала за счет страхового покрытия экспорта, за счет финансирования ставки экспортных кредитов и так далее. Все эти меры уже в ближайшее время должны быть воплощены в жизнь. Мы со своей стороны постараемся учесть как интересы разработчиков и инноваторов, так и учесть и соблюсти разумный баланс между задачами здравоохранения и политикой в области развития фармацевтической и медицинской промышленности. В заключение хочу пожелать всем участникам конференции конструктивной и плодотворной работы. И главное, всегда помните, что бы вы ни разрабатывали, что все ваши разработки и технологии направлены на обеспечение здоровья и безопасности человека. Спасибо большое.
Уважаемые коллеги, следующим на сцену поднимется Майкл Левитт. Майкл Левитт, лауреат Нобелевской премии 2013 года за компьютерное моделирование химических систем. По образованию Майкл сам физик, как и многие из нас. И Майкл расскажет о своем опыте в науке, о том, как его исследования и разработки были применены в индустрии. Майкл, please take your stage.
Hello, everybody. I'm really excited to be here. I've never actually been to MIPT. I've been to Moscow maybe 10 times, and I was involved in a startup company, Algodyne, in Moscow, and most of the people working there were physicists from MIPT who were interested in better chemistry through physics. My background is also physics, but a long, long time ago, in the 60s, I made the change to biology. So I think I can give you some perspective here. I also am at Stanford now, and I think in many ways Stanford is like MIPT. Both universities became much more well-known after the Second World War. Stanford was established more than 100 years ago, but it was only after the Second World War that it started to become a top-level university. And I think there are some interesting parallels. One thing I will say, perhaps at the end, is that making this transition takes time. It's not something that will happen in 5 years. It took Stanford 50 years to go from being an engineering school to being a world-class university with biology and medicine and things like that. It's a very important objective, but it takes time. So the talk I'm going to give is sort of a version of the talk that I gave in Stockholm in December. It's good because it gives the background, and I think the background is important here, and also some of the more modern work that we're doing. So my talk is actually going to have, sorry for that, it's going to have 4 sections. I'm going to tell you a little bit about how it all began, and by it I mean essentially the physics of living systems, because the big discovery that happened in the 60s was that people suddenly realized that biology is all about physics. Something about the birth of computational structural biology, which is sort of my area, something about the current work we're doing, which is the multi-scale dynamics of very large biological systems. These are very tiny systems, sub-nanoscale, but they consist of hundreds of thousands of independent particles, and therefore are large systems. And finally, just give you some feelings of general thoughts. So how it all began. So it really helps to stand on the shoulders of giants. And this is obvious, but it also means that in every field there's like a good time, a magic time. And I was very lucky because I became, well, I guess I was of that age in the 60s. I was between the ages of 13 and 23. And in many ways, the 60s were a time, this is 50 years ago, when people realized that biology is really driven by physics. And the people responsible for this discovery are all very well known. First is Francis Crick, whom I knew very well and worked with, and his paper with Watson on DNA in the early 50s sort of showed how a biophysical thinking could really influence our whole conception of genetics. So this was a really revolutionary piece of work. It was also done in a very interesting way. It was done by a lot of thought. There was a lot of theory. It wasn't theory in the way that we have theory in physics, but it was definitely logical construction. It wasn't simple experimental work. There was also very interesting human dynamics involved. The next person that I want to mention is John Kendrew. John Kendrew, a Nobel Prize winner at the same time as Crick in 1962, solved the first crystal structure of a protein, myoglobin. In those days, there was no computer graphics. Journals never had color. So in fact, publishing in Scientific American was really prestigious because that journal actually did have color, certainly on the cover. So colored pictures were very rare. This is a molecular drawing of myoglobin, of a small part of myoglobin. It was actually painted by an artist. In those days, there was no computer graphics. The really important thing here is that each of these points is an atom, and these atoms all have a particular arrangement. This is a protein from whale. It's a protein which carries oxygen inside whale muscles. Whales keep oxygen in their muscles, so when they dive very deep, they can store more oxygen. And this molecule does that by having an iron atom here and an oxygen binds here. Now, if this is one iron atom, things are placed to a precision of better than 1/10 of a nanometer. So this is sub-nanoscale structure, And every single whale from sperm whale species has exactly the same molecule. So this molecule has a shape, and the shape gives rise to its function. This is a very important message. Both Crick and Kendrew were supervised by the man who invented protein crystallographer, Max Perutz. He is somebody who has a wonderful personality of social engineering. He would work with the cleverest, most outgoing, extroverted prima donna people and manage them all really, really well. So he was really responsible for the laboratory in Cambridge, where I think between people there and alumni, they have almost 30 Nobel Prizes since the 1950s. So it's been incredibly successful, and it's due to this one man who was just a very modest, very clever person. Another person who was very important in my own career is a man called David Phillips, a crystallographer who didn't in fact win a Nobel Prize. He is famous because he solved the crystal structure of the first enzyme. So this went into Scientific American in 1965. That was a very important time. In 1965, I was 19 years old, and it was just very important. He was also a very early supporter of the use of computers in biology, and in fact, in this 1966 paper, there were actually computer models, or at least artistic models of protein folding. So these giants were really important for me because I was growing up at that time. I actually subscribed to Scientific American, but in fact, one of them had an even more important part on my own particular career. So what was the birth of computational structural biology? I was very lucky because I was 20 years old when I got involved in this field, and I was a physicist. I studied physics, I wasn't a physicist, but I also loved computers. So I think today I would be a hacker, maybe I would be in prison, but I was lucky that in those days, in order to get involved with computers, you had to either do high energy physics or biophysics. Both areas, they were computers. So this is the man who really influenced my life enormously. The same John Kendrew that I mentioned. This is him in 1996, a little bit before he passed away. He got the Nobel Prize with Max Perutz in 1962. Immediately, he sat down and wrote a little book called The Thread of Life, which you can actually still get in Amazon, secondhand. But he also converted that book into a BBC television series. So essentially, immediately after he got the Nobel Prize, he said, I have to educate people about the miracle of molecular biology. This is 1964. I was born in South Africa, and in 1963, just 3 days before Kennedy was assassinated, so I remember the date exactly, I came to England to spend a vacation with my uncle and aunt who lived in London. They had television. In South Africa at that time, there was no television. I was completely taken by television. It was a small black and yellow television. There wasn't even white in those days. And on the television, there was a program in the beginning of 1964 by John Kendrew called An Introduction to Molecular Biology, The Thread of Life. And it started out with the revolution in biology, inside the cell, proteins in 1 dimension, proteins in 3 dimensions. All of these topics would still be the basis of a course. Very sadly, the BBC overwrote the tapes to rewrite them, to reuse them, so there's no copy of these lectures anywhere, which is just shocking. It would have been an incredibly historic era. I remember that in the program, they had models, like computer graphics of DNA rotating, and I eventually talked to somebody, and he said there was somebody lying on the floor, physically turning the models around to make them rotate. So John Kendrew, in 1964, I was 17 years old. I had just come to England, and perhaps because of his television series, perhaps because of the political situation in South Africa, which was very bad in the early 60s. Nelson Mandela had gone to prison in 1962. There was increasing repression. I decided that I wanted to stay in England and study there. This program had influenced me enormously. I decided to still study physics. I think I decided to study physics because in physics you have to learn least. It's all first principles. You can sort of guess things. In chemistry you have to study a lot, and in biology you have to study even more. So I guess I decided that I would be lazy and do physics, which I think is a good thing because it's nice to be able to work things out. Now, John Kendrew really was the influence and influenced me to study physics in London. I studied physics actually at King's College in London. There was a Nobel Prize winner, Maurice Wilkins, who had won the prize in 1962 with Crick and Watson. He was the experimentalist who had provided the data that they could have used. In any case, I went there, I studied physics. In my third year, I decided that I wanted to go and do a PhD with John Kendrew. I wrote to him and he said, sorry, no place. So I said, fine, but I have friends in college and they were business people, and they said, if somebody says no, you always say, you come back, you don't just say no. So I wrote back and said, well, it's not this year, what about next year? And they liked that, so they said, come and be interviewed. So I went up there and was interviewed by them. And after my interview, they said, okay, we will tell you in 1 year's time if we will take you. So I said, fine. I went back and my friend said, are you crazy? You're going to do nothing for 1 year. You're one of the best students at the university. You're going to wait a year and then they're going to tell you. So I went there by myself without making an appointment. And I waited outside the offices of John Kendrew and Max Perutz, the man I showed you over here, a very sweet man. And I caught Max Perutz in the corridor and I said, Max, I'm really, really worried. I have my final exams in a few months and I can't study because I'm worried about my future. And he was very nice. He said, come in. And he told me, look, I cannot do anything. I'm co-director with John Kendrew. We have to wait. And I just kept quiet and left immediately. But that was enough. They decided to accept me. And then John Kendrew said he wanted me to go and work in Israel with a very important scientist in Israel. And he then made it very easy for me to go to Israel. I was a 20-year-old person with a BS degree, and he got me a postdoctoral fellowship. It was more money than I'd ever had in my life. It was 4 times more money than I'd been spending every year in England. So I bought fancy suits and tape recorders and skin diving equipment and went off to Israel. So when I got to Israel, John Kendrew had sent me to work with this man, Shneor Lifson. This is a picture taken probably 15 or 20 years ago. In 1967, Shneor Lifson is here. This is how I looked. It's a picture that I like for some reason. Shneor Lifson was he was basically, I guess, a physicist, and he called his department chemical physics, which in fact annoyed all the physicists, because at that time, physicists strongly believed that failed physicists were chemists and failed chemists were biologists, and this arrogance really cost them a great deal. But Shneor Lifson founded the Department of Chemical Physics and not Physical Chemistry. They didn't like that. And in any case, he had a very important philosophical idea. He also had a student that I'll tell you about. But first, I want to reinforce the image, the revolutionary image of biology, i.e. biology is driven by structures. These are protein molecules, DNA. Everything in biology is exquisitely structured. Atoms are in exact places, and they're always the same. They're determined in their places by the genetic information. And because the structure leads to function, this is a very profound, something which is still, everyone understands, but still doesn't deeply appreciate it. Once you have that, it means that physics, the physical interactions between these atoms, essentially determine all of biological function. There's nothing magical. It's very complex, many moving parts, but essentially inside each cell is like a very complicated clock. Okay. So at that time, in 1967, 68, Shneor Lifson was working with Aryeh Varshil. Aryeh Varshil shared the Nobel Prize with me and Mark Harpless last year. And he had a very important philosophical idea that he called the consistent force field. Now, a force field is, as you all know in physics, is an expression for the energy, or the derivative of the energy of the system. In our case, the system is a molecule, i.e. something like this. You can sit down and write down an expression for the potential energy of a molecule like this, and hence the force field. The trouble is where do you get the terms for your force field? Nowadays, you can get them from very expensive, very exact quantum mechanical calculations. You can essentially use approximations to the Schrödinger equation and get the interaction of any pair of molecules at a certain distance apart and at a certain angle. In those days, you couldn't. You had to determine the information experimentally. Now, there wasn't a lot of experimental information. The experimental information was expensive, so it was very important that you didn't have too many parameters. It was important that you had, for example, one kind of oxygen, and that oxygen would be the same wherever it occurred. No matter what molecule the oxygen was in, the parameters would be the same. That was the idea of the consistent force field. Initially, it was basically done for cycloalkanes and N-alkanes, very simple molecules containing carbons. But it turned out very easy, because of this consistent nature, to generalize it very quickly to any molecular system. So here is an example of the molecular potential energy. This is a slide which I guess I hand wrote and then published in 2001, and it's become rather iconic. Just a few weeks ago, it was a party for me in Israel, and the young lady who was giving the party made a cake. And she took the slide and printed it on icing. And this was on my cake. So I was actually digesting the equations in a very deep way. So this is a very, very simple mathematical form. None of you will have any problem. It's a very simple form. It's really very classical. The people involved are all dead physicists. Going back many centuries, you could say it's the dead physicist potential. It's essentially treating a molecule as a collection of balls and springs. These are the springs, these are the springs, these harmonic interactions. These are the non-bonded interactions, the van der Waals interaction between all pairs of atoms, which is repulsive at short distance, attractive in between, and then the Coulombic interactions that can either be repulsive or attractive. Very simple. Now, what's remarkable is that today, 50 years later, people are spending huge amounts of money on big computers that are just implementing this equation. With some changes, but hardly any change. The force field is still what I now call the dead physicist's force field. And this is actually a disgrace, and I will say maybe more about it at the end, because Moscow plays an important part in how this could go forward. Okay, now again, this is unnecessary for physicists, but it's very important for other people to realize that once you have an energy function, i.e. this function, just to remind you, some of these things are geometrical. B and theta and phi and r are geometrical parameters that are derived from the coordinates of the system. It's very easy to get the forces by taking the first derivative of this function. These values, the B0, the theta 0, are the force field parameters that were determined in a consistent way by Lifson and Warshall. So they basically found a way to get approximate values for all the parameters in this, and there are quite a few parameters, probably a few hundred parameters. But once you look at them, you see that they can be guessed. Almost all bonds are the same, single bonds, double bonds, it doesn't matter, and so on. Okay. So once you have the energy function, you can now do all sorts of things. You can do energy minimization, which is to drop down into the first local minimum. One of the nice things about all of this, it generalizes from one dimension to n dimensions very easily. You can do normal mode dynamics, where you basically solve the vibrational frequencies of the system and see how it moves about a minimum. Then you can do molecular dynamics, where you essentially do minimization with kinetic energy, and you can let the system propagate like that. If you don't take the energy out, it just keeps on going, and you can use up a large amount of computer time. Certainly a few years ago, the majority of American computer time was used on this particular equation. So it's quite amazing, on molecules. And finally, we have the Monte Carlo method, which is difficult to illustrate in one dimension. This method probably has the most interesting history. Just something about history. You could probably say that minimization goes back to Euclid, but probably goes back to the ancient Egyptians. You can attach names to, say, Galileo to a pendulum, Newton to dynamics. And this is the most interesting one. This is the Monte Carlo method. The first author of Metropolis, who was born in 1915, but the last author of this very famous paper from 1955 is Edward Teller. So this method was really developed for neutron diffusion in hydrogen bombs. It's kind of interesting, because in some ways it's the most simple of all the methods, but it requires computers. Okay. So once you have an energy function, we can apply it to molecules. And again, this word multiscale was a word that I never used, but the Nobel Committee used it. And I think they used it because they wanted to give a Nobel Prize for more than one thing. But you can't give a Nobel Prize for more than one thing. So the one thing was multiscale models. But it's like saying, we'll give you a Nobel Prize for a basket of discoveries, one basket of discoveries. Anyway, I was very happy, obviously. But it is interesting because in a short period, we actually did develop multiscale models. Now, what is it about multiscale? So essentially, it goes back to a quotation which has been attributed to Einstein: everything should be made as simple as it can be, but not simpler. And that sort of seems obvious, But it's very important when it comes to doing computer calculations. If the calculation is not simple enough, then it will take forever to do. And remember, computers were very slow in the late 60s. But if it's so simple that the results are meaningless, then forget about it. So people say, you know, the physicist says, we would like you to model milk production. And he says, well, let's start with the spherical car. Now, a spherical car would probably not be useful. But maybe a car made from 10 spheres would actually be very useful. So one needs to choose the right level of simplification. And that's critical. So at about the same time that I was in Israel, I was actually being a programmer for Varshil and Lifson, writing their programs for small molecules, but I also volunteered to actually build a physical model of the enzyme lysozyme with Yuval Eshdat, who was a PhD student of Natan Sharon. Natan Sharon was working on lysozyme as a biologist, and he'd obtained from David Phillips the atomic coordinates of lysozyme on paper, and I volunteered to build this model, which took about 3 weeks. Very difficult. It's very hard even to see what's going on, but essentially you have 101, even more, maybe 1,500 different vertices here, and you have to build a model where everything is placed quite precisely, probably within 1 centimeter accuracy, even better. It's a lot of work, and once you've done it, you really want to computerize it. Now, we had been working on small molecules, but I suddenly realized that the same programs that could work on small molecules, calculating vibrational spectrum, which requires a matrix, a second derivative matrix, could actually be used to minimize large molecules. So I took the coordinate listings of lysozyme and also myoglobin, gave it to the typing pool at the Weizmann Institute. They typed the punch cards with all the coordinates. They actually typed them twice to see if there's any errors. Then I put them into the computer, and this led to my first paper in 1969 on protein energy minimization. Essentially, we took this force field, put it into the computer with the coordinates of myoglobin and lysozyme, and minimized them. The important thing is that the minimum was stable, which is not completely surprising because the minimization was not that effective, but it was essentially the first time an entire protein had been dealt with as a single physical object. I then went back to Cambridge, did my PhD, not in fact with John Kendra because he had left by that time, but with Bob Diamond, a crystallographer, and then came back to the Weizmann Institute in 1973. In my year in Israel in 1968, I'd actually got married, and by this time we actually had 2 children already. So my wife had been working very, very hard. She also actually had to leave the house to work, and as a result, I can never work at the laboratory. I can only work at home. So nowadays, I only work at home. I come to the laboratory to drink, to eat, to have parties, but I work at home. So this led to a paper in 1975, again with Ariel Varshil. I cannot move that point, so I'm sorry, it doesn't move. And I'm not targeting with my red laser, so he would not be happy. So in this work, we basically took a protein and simplified it. We simplified it by leaving out most of the atoms. Now, that sounds like a very easy thing to do, but when we had to do that, it seemed like complete sacrilege. How could you leave out atoms? So let me just say why we did this. In the early 70s, there were many space missions. There was the lunar missions. There were astronauts in space. And at one point, Ari and I were joking about we would like to simulate protein folding by going into a space shuttle, because the models are very heavy, and if you try to move the models, the gravity affects them. But of course, that was impossible. So we said, okay, let's on the computer simulate what it would be like to be in a spaceship with these models. And if you do that, it's a model of a model, who cares? So we left out all the atoms, had a very simple method which allowed us to fold a protein by a combination of minimization. When you get to a local minimum, you then have to disturb the structure, and we did that using normal mode jumps from local minima, where we found the curvature, we moved along a line of least curvature, very physical technique, and escaped from the minimum. This ended up folding a protein in maybe 20 minutes of computer time back then. Of course, the answer was not very good, but we thought it was better than it was, but it was the first computer attempt to fold a protein, and in fact, these simplified models, which were actually called coarse grain models, we just called them simplified, have now become very much more popular. At the same time, Ari and I did something else that was probably even more important. We were trying to understand how the enzyme lysozyme works. In 1965, when David Phillips had published this, he had hypothesized that lysozyme, which is an enzyme that breaks a sugar molecule on the surface of bacterial cells, does so by straining the molecule, putting a force on the molecule and making the molecule break by physical force. Now, that's a nice idea. The trouble is that when I did calculations during my PhD on this, the enzyme lysozyme, although it's made out of atoms, is too soft. It's got too many degrees of freedom, and I always say it's a little bit like trying to break somebody's back by putting them on a very soft bed and pushing down on their legs, and you can't do it. Put them on a hard bed, it's quite easy, or a piece of wood or a piece of metal, but it's really impossible on a soft bed. The enzyme is too soft. So how does the enzyme break the system? Working with Arya Varshal, we decided we needed to put in quantum mechanics. Of course, every time you have quantum mechanics, you have to worry about every electron. So now you have a lot more degrees of freedom. These are the orbitals. So what you need to do is treat a small part of the system by quantum mechanics and all the rest of the system by classical mechanics. This became known as a method of QMMM. You'll see that I'm not very good at names. We just called it, I don't know, mixed models or whatever. I even forget the title. And this ended up being a very popular method which is used more and more and more. There are certain interesting tricks about how does the quantum part see the classical part, and that's usually through electrostatics. How does the classical part see the quantum part, and so on. But this method is now used in many, many simulations of reactions. And again, I think we need better methods. Okay, so the word spreads. There was a very important paper by Martin Karplus and his co-workers in it's not 75, it's 77, I'm sorry. Okay, it isn't 75. I must have copied this from my previous slide. Okay, anyway. So in 77, the first simulation of protein dynamics, this was a small protein, bovine pancreatic trypsin inhibitor, which had in fact been solved from Robert Huber's lab. I think that's how we met in the early 70s. It was a very important protein because it only had 1,000 atoms, whereas all other proteins had several thousand atoms. It was a very small protein, and Karplus had simulated the dynamics of the system. He had run dynamics for 9 picoseconds in vacuum. It required a few hours of computer time. It was a very important thing. But the calculation had been done in vacuum, and real proteins, their medium is very important. So I then spent some time, but let me just say that one of the hard things about dynamics, it was a great deal of data. You get a snapshot every femtosecond. So movies are really important. This movie is the first movie ever made of protein dynamics. It was made in the late 70s by somebody, Richard Feldman at the NIH. Each frame took many hours of a really expensive computer. The people who actually did the graphics went on to found the company Pixar. And computer graphics, they were very good graphics experts. Now, when you look at the motion, it's actually interesting because it's not moving around like you might expect. It's sort of random motion, but it's also bounded. The atoms are moving around, but they're bumping into each other. In particular groups, this is a water molecule escaping from the system. Everyone focuses on that. But what's much more important is to see how it's moving, but it stays the same. And that's what I think is essential about most of the motion in biology. Again, this is a system without the water. So I then went on to worry a lot about the water. It took more than 10 years to go from simulations in vacuum to simulations in water. The reason is that the waters add something like an extra 9 times more atoms. But more difficultly, the energy functions you use for the protein have to match those you use for the water. And this involved using charges and electrostatics, and it was a difficult calculation. It was actually done together with Ruth Sharon, a scientist at the Weizmann Institute. This shows the protein with a few waters around it, but in fact the protein was in a box. And the important finding is shown in this graph that if you run the calculation in vacuum, it moves away from the X-ray structure. If you run it in water, it stays at the X-ray structure. This calculation actually used up $200,000 of Cray XMP time, which was a very fast computer in the 1980s, and it cost the NSF more than $200,000. So that was a very expensive calculation. But in fact, since then, all calculations on proteins are done in boxes of water. So this ended up setting the protocol that used. There were certain other details. If you do the calculation in water, you include all the hydrogen atoms on the protein because it makes very little difference, and so on. Okay. Here's an example of a movie of part of a system like this. It's just the alpha helix in a box of waters. I think it's moving. Yeah. So here we have a simulation of an alpha helix in a box of waters. I actually made this movie in 1990. It was made by photographing from the screen, and I didn't even line the camera up properly. This was an important movie because I actually showed it to Linus Pauling, who had invented the alpha helix in 1951. And I guess he was impressed. I don't know. But I was very impressed making it. This just shows that this is the helix at room temperature, 5 degrees centigrade, and it's basically stable. But at high temperature, the helix is unstable. That's just common sense. But common sense is very important. When you do calculations like this, common sense is much, much more important, even before you test against the experiment. So this structure is unstable, it comes apart, and there are interesting things about how it comes apart. Okay. So now let me move to the third part, which is essentially ongoing and future work, just to give you a flavor that one is still doing things. So an important lesson about modeling large systems is to reduce the degrees of freedom. You don't necessarily have to throw away the atoms, but you need to treat parts of the system as rigid. And the example that I always give is that when I move my hand, I need to do the calculation about, I don't know what an arm weighs, maybe 5 kilo, how many atoms there are in 5 kilo. And I guess if you can divide very quickly or multiply very quickly by the molecular, it's probably something like 10 to the 27, I would guess, maybe 10 to the 26. 10 to the 27 atoms in my arm. When I move my arm, I don't need to simulate the motion of all those 10 to the 27 atoms. They're all going to move in a way that's bounded by very simple degrees of freedom, which might be a few pivot points. You can probably define the motion of a hand completely by 10 degrees of freedom, or at least an arm. So choosing degrees of freedom is very important. Sorry. The first thing I wanted to show is a simulation actually using something called a Markov state model. I'll describe that during the motion. This is a large system, almost 500,000 atoms. This is the system that Roger Kornberg got the Nobel Prize for 7 years ago. This shows the motion of that system, but this motion is not simulated by molecular dynamics. It's simulated by making a state model of the entire surface and then doing random jumps between the states. So this is a much easier way to simulate very large systems. It was worked out together with Vijay Panda and others. This is an enzyme that basically takes DNA, shown here in blue, and copies it to RNA, shown here in red. And the key thing we're going to look at is one very small part where this nucleotide, one base of the DNA, moves from being sitting over here to sitting over here. This is crucial for the function. So again, details really matter. It starts to move. Here you can see its progress. It jumps part of the way over there. It stays there. And now it's going to jump all the way. And it will jump back again. So again, everything is very Brownian. Everything is very random. There's nothing happening. It's deterministic in an average way. Statistics is really important. Another system are normal modes of ribosomes. This is a calculation that involved quite a lot of difficult programming, diagonalizing very large matrices and worrying about their conditioning number and things like this. I still write programs myself, and I was very pleased to have got this to work. This is a system with, this is the ribosome, again another Nobel Prize a few years ago. You're not Tom Stites and Venki Ramakrishnan. And this is a very large system which is actually used in the body to make proteins. And here are 2 transfer RNAs shown here in cyan. And these motions are just simply the normal modes, exaggerated in size, but the motion is very suggestive. It suggests how this might function. This is the groove in which the messenger RNA sits. People have already seen this, some of these motions by EM. And this is a great way, and again, because this is done with all the atoms, we know exactly how every single atom moves during this motion. This is some work that was done by an ex-postdoc who's now in computer science at Oxford. He was working on nucleosomes, and the idea basically here is that can we predict along some DNA where the nucleosomes will bind. Nucleosomes bind by the DNA twisting around them. The DNA has to bend easily. And by a calculation that was completely dependent on atom potentials, we were able to get a pretty good agreement for when nucleosomes bind. That's difficult. Up till now, people had done this by knowledge-based models using experimental work. And again, there's a tension in this whole field between knowledge-based models, i.e. based on experiments, and physics-based model. There's a lot of interest nowadays in big data, and all big data is essentially model-free modeling. You can model the shoppers in a store, and that isn't enough. Okay. Here's some more recent work on other systems. This is a molecule called the chaperonin. It's a very interesting puzzle because this is a molecule that has there's 2 rings, 1 ring here and 1 ring here. Each ring consists of 8 molecules that aren't identical, but are almost identical. And this makes it a very difficult structure to solve because you don't know where to put them. This had been looked at by electron microscopy and by crystallography, and they got different answers for the arrangements. There are actually 8 factorial ways of coloring an object like this. We then decided to look at this using mass spectrometry, which is a very powerful technique, where you look at crosslinks. These show some of the crosslinks. And using this crosslink, we could actually determine what the structure was. We also then used a new method of crystallography to actually determine what this would be crystallographically. And this is interesting. Essentially, what we did was to do brute force. We did something which any crystallographer would say was impossible. We took this object, which consists of 130,000 atoms, and built 2.5 million different models of that object. And then took each model and calculated the agreement of the initial structure with the X-ray data. Now, the X-ray data is something called an R value, which is a measure of the agreement between the calculated structure factors and the observed structure factors. And essentially, for random structures, you get 50%. So when you look at the values, all the values are shown here. This is 51%, 49.9%, 51.1%. Everyone would say these numbers are random. But when you calculate all of them and look at them very carefully, what you find is that this distribution, shown over here, and in green on a log scale, is not random. A log scale would be perfectly parabolic, but there are additional values over here, and when you calculate the best value, it's actually 10 standard deviations from where you would expect it. 10 standard deviations is a long way. But then if you actually just take the, say, 10 best values over here and line them up, and it's important to realize that this structure can be defined by just saying in what order the colors are in each of the rings. So essentially, it's a string of 32 letters which define the color ordering. Each structure has a different string. You can take all the strings of the best ones and just average them, and out comes the right answer completely unambiguously. It even works on a less good structure. We then went on to use the mass spec and the similar method together with Roger Kornberg on a very large complex called the pre-initiation complex, which had been solved by, I would say, old-fashioned EM, which is low resolution. There is now a revolution in EM, and you guys should buy yourselves some new microscopes here because it is amazing. This is what old EM looks like. You have this blob, and you have no idea where the different groups should be. You know inside there are different factors. Each ball is about 300 amino acids. So now we're saying a protein is a single ball. That was enough because you can put them here. You can calculate for all different arrangements of this what the mass spec would be, and you find that there is only one that agrees very well. That one is then found to actually give you a very consistent model which explains exactly how the DNA fits into here, and this led to a recent paper which was fun to do. Okay, so obviously applications to human health have been talked about, and again, in talking about applications to human health, I have to say it takes time. This example is an enormous success, but you had to wait 25 years. So this is an example where we want to make antibodies to treat cancer. This is an idea which was around in the 80s. I had worked on purely theoretical work on how you would model an antibody. Antibody is a protein that has 6 loops that have a lot of variability. Those loops are responsible for the antibody recognizing any foreign object that comes into the body. They're basically our defense forces. And these loops let the antibody understand what they're doing. I had written a computer program in the 80s that would allow you to model any antibody structure. I went to Stanford, teamed up with the group that had been formed, a new company in 87, PDL Biopharma, and they wanted to basically turn this into a business. Now, they succeeded, but it took so long that the company didn't exist anymore by the time they succeeded. But what they did do, we published this paper in 1989. At the same time, Carrie Queen, who was the lead inventor of the company, also patented things very, very well. So he wrote a really good patent describing on how you could model antibodies. Now, we weren't the first. Greg Wint in Cambridge had actually humanized antibodies, but his method was not sophisticated enough. We found that by modeling the entire structure, you could actually improve things. Anyway, the Queen patent held up. It's about to expire, so this is now 25 years afterwards. But it turned out generating an enormous amount of revenue for the company. This is the revenue to the company in $1 million, so this is $400 million over here. These are the names of some of the antibodies, Herceptin, Avastin. These are very famous names of antibodies that really help people with certain cancers. Now, the real money was not, this is money from the license. This is 5% or 4% of the money. The real money was made by a biotech company, Genentech, which is now purchased by Roche a few years ago, and they must have been making $20 billion a year, $20 milliard a year from this. Now, of course, they spent a lot of money. I mean, this whole project probably cost at least $100 billion. This is a lot of money. And it's just realizing that these things take a lot of time and they require a lot of money. This is just a picture of what I'm currently working on. This is the important one. Ivan Ufemtsev actually graduated from Institute of Physics, and I guess Yana Goffman is also a Russian-Israeli. My group is very small. Right now, Ivan is trying to solve the X-ray phase problem, a well-known impossible problem. We're doing other things. Andrea is working on methods to compare whole genomes, and Yana Goffman on how you do EM of membrane proteins. But basically, we're doing a lot of things with a small group. Just finally, some general thoughts. There is no doubt that the work we did was pushed ahead by technology. And to put that into perspective, computers have changed. They are 10%, they're 10,000 times cheaper, 10,000 times faster. They have memory, which is 10,000 times more, and the size is 10,000 times smaller. Now, I'm actually surprised why each of those numbers are so similar, but they're only accurate, probably a factor of 10. So maybe they're much more than, it depends on the size of the hole and whether the laptop is open or closed and whether you have to worry about the heating and so on. So the size is a hard thing to define. But it's very easy to put these into terms of a motor car. And this is obviously done for Sweden. If a Volvo would cost $3 an hour, would have a top speed of 1 million kilometers an hour, would hold 50,000 adults in comfort, and would park in a shoebox. Now, that is really true. I mean, computers have become that much more powerful. And this means, and the fact is that if now I would very easily bet that we will look forward in 50 years and say, gee, it's another factor like this. I mean, there's no doubt that we're increasing all the time. People are now using multiple processors, gaming machines, GPUs, etc., to get enormous speed-ups. Family support is very important for any scientist because scientists basically are married to their work. And if they want to have a family, they've got to have 2 wives, or at least one, but a very good one. This is my mother, who's actually still alive. She's 99 years old. My wife of 46 years, and my cutest granddaughter. We actually have a lot of grandchildren. My wife said it wasn't fair to put her in, but it's sort of a picture of my wife. This is not a disease. She's decorating herself for some, like Halloween. You know the old saying, behind every successful man there is a surprised wife. This was actually sent to my wife just after she'd heard about the Nobel Prize from a PhD student in Stockholm, whom we knew. And I guess it must be very well known, but it still catches a laugh almost every time. Advice to the young. This is where I really care, because I think that young people are not given as much independence today as they were 50 years ago. When I did my PhD, I didn't write a single paper with my PhD supervisor. We actually, at one point, had related papers that were in adjacent pages of the same journal. Max Barutz, who supervised Francis Crick and John Kendrew, did not have his name on their papers. So this is a very important thing, because young people, I mean, innovation comes from young people. You just look around at the computer industry, You have to drop out of university to find Microsoft or Apple. You have to drop out of your PhD program to found Yahoo or Google. They are in such a hurry to do new things, they can't wait until they're 22. They've got to start when they're 21. So I think we need in science to realize that young people need to have more independence. You have to be persistent. If they say no, you've got to believe in yourself. You've got to be very stubborn. One of the good things nowadays is that because of the internet, there's no such thing as bothering a person. Everybody is so bothered by so many spam emails. If they don't answer you first time, send it again. Like right now, I don't answer any person I don't know who hasn't written to me 3 times. It's just a filter. So don't give up. Believe in yourself and don't give up. Be original. Try to think about for yourself. Everyone says to me, what would I tell young people to do now? And the answer is think about it yourself, because if I tell you, other people will do it. Try to think about something which combines your interests, your passions, your background with the problems. There are an infinite number of problems in biomedical sciences. Biology is so complicated that we aren't even near understanding any of it. And finally, be kind and good to people. It doesn't hurt. You don't have to push in lines. You can just way back and just take it easy. Be a geek. I want just to thank my towering heroes of science, as I call them, Schneur Lifson, who was the person who really got me into this, John Kendrew, Max Perutz, Bob Diamond, who was in fact my actual PhD supervisor, Francis Crick and Aaron Klug, who were all very influential on me in Cambridge. My group, which was not terribly large, but I have a very wide variety of different kinds of people, and it's been fun working with people. For my first 20 years of my career, I actually worked by myself or with colleagues. And that's not a bad way to work. It actually works pretty well. You don't need a large team of people. Teams can be a cost as well as a benefit. Finally, I want to thank the Nobel Committee in Chemistry, and that, obviously I want to thank them. They gave me a Nobel Prize. I mean, what else? But the fact is they did something that was very, very brave and original, because they basically gave a Nobel Prize for computers. Now, everyone knows you don't give a Nobel Prize for computers. There was a Turing Prize, there's other prizes. Who gives Nobel Prizes for computers? Not only that, but they gave a Nobel Prize in computers for work that was mathematical and physics on large molecules in biology with biomedical applications that they emphasized very strongly. So they went all the way across the disciplines, and when I spoke to them, and that's one of the good things about you meet the group, you talk to them, they really felt that they were going to be increasingly looking at these multidisciplinary approaches. Although the categories in the Nobel Prize are very rigidly defined, they do whatever they want. This is really good for the field of computational anything, and the future is definitely computational, whether it's your phone or your GPS, or almost anything else you do. There's no doubt that computers are going to be very, very important. And this is sort of a joke, because just after the award was won, I was on the American football field at Stanford. This is at the 3/4 time of a game, and we were asked to come out on the 10-yard line, the Stanford 10-yard line. Stanford was winning at this time. This was at Stanford. This is my 10-year-old, 11-year-old grandson, and at this point, 50,000 people had stood up and were shouting, "Nobel Prize, Nobel Prize." People asked me what was the most exciting time, and that was pretty exciting. Very shortly afterwards, my grandson jumped on my back, so it was very exciting. And finally, I want to thank you all for your attention.
Michael, thank you so much. I would like you to have a seat, and then I would like to invite our other guests. First of all, Robert Huber, who is a Nobel Prize winner, on the stage. If you don't mind, could you? And so, if you have any questions to Michael, please, you could come here and ask him directly or with a translation, as you wish. And I also would like to invite Andrei Vashchenko on the stage to have a seat, and Alexei, and Evgeny Kuznetsov from Russian Venture Company, for a small discussion and answering questions from the audience and from myself, if you don't mind. And so while everybody is collecting on the stage, so my question will be, the first question to Michael. Actually, the same question for everybody on the stage. So, from your point of view, what are the most promising technologies of the 21st century or maybe the next 10 years, and what kind of elements should have any university that want to be competitive worldwide?
So normally I would not answer a question like this. I would say we don't know. But it turns out that one area that has actually improved enormously in the last few years is electron microscopy of large molecules. It turns out that when you shine, this is all just physics, when you shine an electron beam into a molecule, the energy you deposit is quite large. And in the first instant, the molecule jumps, it moves. And as a result, it's like if you photograph somebody and when you flash, everyone jumps. It's going to be blurred, especially if you jump quickly. So it turns out that they now have a method of collecting the electrons very, very quickly with very sensitive devices, and they can collect 100 frames a second. That way you can actually see the movement, you can correct for the movement, and now people are getting resolution of electron microscopy, which is 3 or 4 angstroms, whereas it used to be 10. So suddenly electron microscopy, whether it's on nanomaterials or anything, is going to be much, much more precise. I think this is a big, big breakthrough. There are many different techniques. I think that in general, mixing up microengineering, silicon-type engineering. I mean, there are now laboratories on chips. Mm-hmm. This is another very important area. I think that physics actually has a huge amount to give. But, you never know what the next big thing is going to be, except computers will be involved. But computers are it's like saying, papers will be involved. It's a medium, it's not a method.
Okay. And so, Robert, I just repeat the question that I have for everyone. So, from your point of view, what are the most promising technologies of next 10 years will be, and what kind of element should have any university that want to be competitive on the world stage?
Well, let me first say the wonderful thing with research and science is that it's not predictable. The revolutionary new ideas come out of the blue sky, more or less, like lightning. And we don't know what it will be. But one can make some prediction. I would agree with Michael that computers will play a major role. Of course, the final goal that in structural biology we have is to predict ab initio macromolecular structures and interactions. There is enormous progress in that field, but not yet at the stage that we can that we can rely on them in that sense that we use these predicted models for, let's say, drug design and drug development. So we still need to do the experiment. On the other hand, protein folding and protein-ligand interaction happens in our cells millions of times while I'm sitting here. So there are rules, and we should be able to find them precisely enough to do a reliable prediction. Whether this will come in the next 10 years, I'm not sure. It will need computers, that's quite clear. It will need bright young people. It perhaps will need a novel idea. Maybe the interactions that we have now, which Michael described, are not sufficient. Maybe we miss some important interaction. Maybe we need an Einstein of biology with a bright new idea and approach. I don't know.
Okay.
But it will happen.
Thank you. And I know that you travel a lot and you work with international groups, so in a lot of universities, and so from your point of view, so can you give an advice, for example, for MIPT? So what kind of infrastructure should be built to be successful?
Well, I strongly believe that structural biology will play a major role in understanding biology and making use of it. This is crystallography still. It is electron microscopy. There is enormous progress in the last 5 years or so, driven by the technological advances. There is an MR, but I think most of the breakthrough progress will come by electron microscopy, often in combination with X-ray crystallography, whereby we look by EM at the large aggregates, dissect it into its components, which we can crystallize, and then model back. So this is what I think you should have here. The Institute has a strong physical basis in history that would fit perfectly.
The same question maybe for me to Evgeny. So you are director of project office of Russian venture company. So how could you comment? So you see a lot of projects that come to Russia or to RVC. So and you also communicate with a lot of international partners. So from your perspective view.
Спасибо за вопрос. Довольно уверенно можно сказать, что завершена эпоха, в которой было определенным образом картированы и технологии, и науки, и мы существовали в рамках определенных представлений о том, в каких сферах можно добиваться какого-то успеха. Сейчас практически все наиболее крупные результаты, как в технологическом плане, в бизнес-плане, это всегда конвергенция технологий. Это всегда что-то, родившееся на стыке наук или на стыке технологий, или на стыке типов деятельности. И ключевой, конечно, рецепт для любого и университета, и любого бизнеса, и любой компании, и любого региона, кто хочет добиться сейчас какого-то успеха, это осуществлять конвергенцию. И в этом смысле именно университет может и должен быть ключевой инфраструктурой, где происходит на самом раннем этапе взаимопроникновение довольно разных сфер знания с тем, чтобы выстраивать современные представления об их взаимодействии. В России мы сейчас немножко отстаем именно в плане понимания вот этой современной основной динамики. Даже вот такое направление, как биоинформатика, в котором там биология и IT переплелись, да, не развлечения, у нас по-прежнему, в общем-то, отстает. Это множество других направлений. Я думаю, что ключевой рецепт и ключевая задача, которая перед нами всеми стоит, это выходить за рамку старых парадигм, выходить за рамку старой системы, вплоть от классификации до системы распределения знаний и создавать вот это взаимопересечение. В бизнесе это происходит непосредственно, и тоже касается, кстати говоря, стиля деятельности. Уже нет классических ролей ученого или классических ролей промышленника и так далее. Необходимо очень глубокое взаимопонимание, очень глубокое перемешение практик. Необходимо не только понимать другого человека, но и действовать как он, действовать, иметь возможность действовать его правилами с тем, чтобы заходить на его поляну. И кроме как в университете, этому негде ни научиться, ни сформировать первичные практики, ни начать так действовать, ни создать те новые бизнесы и те новые виды деятельности, которые уже в таком качестве начинают продолжаться. Я думаю, это, наверное, главный рецепт сейчас. В МФТИ, наверное, даже проще, потому что есть достаточно богатый опыт, но в целом мы находимся только в самом начале движения, которое везде уже достаточно далеко продвинулось.
Thank you. I just translate very shortly. So Evgeny told us that he thinks that so we should go beyond the frames of the some ordinary classical frames of teaching, studying, even so scientific directions, and convergence and integrations of different fields of knowledge like biology and mathematics, biology and informatics is a kind of advice for success. Alexei, could you comment something on the same questions?
Please, Mr. Levitt. Okay, please. All right. Yeah, I have a few comments from a macro level, from a strategic level, I should say. If we take the Russian healthcare structure, government spends a lot of money on healthcare. And as we see, if there is a new molecule coming out, like Avastin or Herceptin, government should spend a lot of money on that. So the government task for the next few years is to make those spendings lower, to make medicines more affordable, and thus make more people benefit from it. That is why maybe treating a disease sometimes costs more than predicting a disease. So from the macro level, I think that we need to develop more technologies that would predict a disease, so diagnostic technologies. And that would make people healthier, and that would prevent them from having a serious disease that would cost a lot of money to treat. So that's my answer.
Thank you. Michael, you need...
Yeah, so I guess I'm unusual because I've actually worked for almost 10 years in 3 different countries, in Cambridge, England, at the Weizmann Institute in Israel, and at Stanford. So I've actually, I'm quite good at comparing different places. And I think if I ask what are the key ingredients for making a Silicon Valley, or whatever you'd like to say, I think that one is that there needs to be, I'm not going to say necessarily academic freedom, but for example, if I was in an institute and I wanted a new laptop, if anybody told me what laptop to buy, I would leave. I need to be able to take my credit card, buy a laptop, and be absolutely sure I will be reimbursed. In Cambridge, I would probably be given one from the stockroom. They would have them waiting for me. In America, I can buy it on a credit card. That kind of bureaucracy is killing science. Francis Crick once said that if he comes to a new laboratory and he sees the corridors are clear, it's a really bad laboratory because it means that the fireman is more powerful than the top scientist. Be very careful of that. The second thing is that one needs to actually combine with the physics and the computing, the humanities, and medicine. I would say that MIPT is probably like Stanford was in 1960. In 1960, a medical school was brought to Stanford. It takes a long time. I think it's really important for government to realize that this is a 50-year investment, not a 5-year plan, but a 50-year plan. It'll happen, but you have to be very patient. The other thing, and this is something which is maybe difficult, I'm not sure if it's certainly difficult in Israel, difficult in England, not difficult in the United States, you need to let faculty make lots of money from connections with companies. Silicon Valley was created by the venture community, but equally well because the scientist could, on his 1 day a week of consulting, make $1 billion, and no one minded at all. No one said, oh, but how can he be a good scientist if he's making $1 billion? Now, I didn't do that, but there are many who did. Google started out as 2 Stanford PhD students, and so on. So it's understanding that you can get academic conflict of interest taken care of, but you don't have to make people poor. And the fact about venture communities is, in the end, also, you need a very liberal licensing of technology. Many people think now that you make money by licensing the technology and then giving it to a company. No. You give it to the company for free, and then you make money when they build on your land or they come to your shopping center. Stanford gets more money from its shopping center than it gets from its technology licensing. These are not obvious things, but these are the things you need because starting a Silicon Valley, you have to be very careful and very delicate. Just one answer to your point. I think that what is going to be critical for human health are smartphones. My phone will call the doctor and say, you know, he's not walking so steadily, maybe he's getting Parkinson. It's very easy for a phone to do that. The phone could be monitoring what I'm eating. It could be monitoring my blood pressure, etc. Even monitoring what I'm saying, you know. He had a big argument with his wife and his heart started beating, you know, he needs more medication or whatever. So I think that this is going to be the big revolution in healthcare and it will save a lot of money. And again, this is where the new ideas have to come. But I think it's really important to encourage, you know, I think if you want to look for models of innovation, there is no doubt that the most innovative field right now are the apps for the iPhone and for Android. There are such innovative applications because it's open. Anybody who wants to develop an application can develop one. And they're amazing. And some of them lead to amazing successes. Some of them don't. But I think this is an area where we have to try to get our science to go.
Robert?
Well, a remark I would like to make in support of basic science. Your question to me was what breakthrough discovery do we maybe expect in the next decades? I said we don't know, but what is quite sure is that it will happen only if basic science is supported at the universities, so that new ideas can develop in the young brains. So this is quite essential. Support of basic science.
Thank you. And Andrew, from your side, any comments?
Thank you, Alec. I already make a presentation, so I believe I already tell what. Maybe we give the people asking the question to our guest and give more time for the question from our colleagues.
Есть у кого-то вопросы из зала? Давайте, можете подойти. Камиль, я первый вижу твою руку.
I have a question to Michael and Robert. What do you think? Is it going to be possible to be able to model and simulate the whole cell soon and eventually to be able to substitute the experiment?
The issue of whether it's possible to model a whole cell, I'm sorry, forgive me. The question was whether it would be possible to, I guess, model a whole cell, and you know, I don't see any reason why not. It may be as difficult as modeling a whole economy or a whole city, but there doesn't seem to be any fundamental reason. I think the question is going to be, you know, what would a model of a cell do? And I think one explanation would be that you could ask it questions like, if I cut down the supply of oxygen, what would happen? And it would actually give you the correct answer at any level of resolution, because one answer would be the cell dies. But the other answer would be that a certain protein was upregulated, another protein was downregulated, and certain processes kept on going. So I think sometimes I would say that a really good model of a cell would be like a cartoon. In computer graphics now they can model people. But the trouble is all those models are not really physical models. When they have a model of water in computer graphics, it's actually artificial. It's not real. They've learned how to model what water looks like, or modeling cloth. Some of it's physical, but I would say it's actually interesting that a lot of computer gamers know a lot of physics nowadays, because they've got to know about Newton's law and transformations and things like that. But I think I think games are very important. I would love to see, maybe a cell simulation would be like a game. You would put on your three-dimensional glasses, you would go into the cell, and you could actually make certain things happen. But remember, a cell is a very complicated object. A cell is probably as complicated as New York City, maybe even more complicated. So I think, but I see no reason why it shouldn't happen. It's a great frontier.
Is there any other questions from the audience?
Alex Schneider, Boston. Thank you very much. You just gave an excellent example of an evolution of scientific field over the course of half of a century. And I think every discovery, every Nobel Prize, every great inventor of last 50 years came because of two major inventions of American system of scientific metrics and system of grants. However, a package of a first mover advantage has another side of the coin. America, in addition to great advancements which humanity has due to these two inventions, system of scientific metrics and system of grants, we were also the first country to run into disadvantages or shortcomings of these systems. Now, there are two types of idiots in Russia. One type of idiots say it's a foreign system, we should ignore it. Another type of idiots says they had great advances, so let's copy it, carbon copy it per se. So would you please give a word of warning, not where this system could be used, because this is more or less obvious, but where this system has to be critically analyzed and comprehensive analysis, what should be avoided? Thank you so much.
So that question is, I could not have imagined a better question, because in the last few months I started to examine, I like statistics very much and I like numbers, and I love to play with almost any program that there is. And I started to look very carefully at the statistics of the American National Institutes of Health. This is giving out something like $30 billion a year for medical research. And people are saying that the enormous driver of the biological revolution in the United States over the last, probably since the early 50s, was because of the NIH. Now, what has happened is there's actually been a disaster without anyone realizing it was happening. And that can be summarized very easily by saying that in 1975, the average age of somebody with an NIH grant was 40. Today it's 53. It's increasing at a third of a year per year. So in about 50 years' time, all NIH grant holders will be dead. Now, the reason this is happening is that the system is not controlled. The NIH system, nobody has a quota that says such a fraction below 40 should get. It's a system that I say is peer-reviewed, where other grant holders are reviewing the grants. The trouble is that the peer committee is also getting older by 15 years. And when you ask somebody who's 60 what is young, they say, oh, 55 is really young. Young is 20, and maybe 25 is still young. So the system needs to be changed. It's also become a big problem because the whole system of independent groups means that each person has like 50 children in their research career. And that kind of expansion is crazy. We don't have enough need for scientists, certainly maybe worldwide. But the USA is saturated. It doesn't need more scientists. There need to be models of science where not everyone is independent, not everyone has their own group. There needs to be more cooperation. Again, I didn't go to the United States until I was 40 years old. And for the first 20 years of my career, most of the work I did was either by myself or in collaboration with somebody who was generally of the same level as me. And that is a very powerful model. And again, if the two people, now they say, oh, we cannot judge him by a metric, because if he has anyone else on his paper, then who did the work? So you need to collaborate with somebody who is very different. But that's also a good idea. So I think the United States system is good for those reasons you mentioned, but as exactly you said so well, it's also bad for those reasons. I think if there was infinite money and the world was infinitely large, then it would be a good system. But right now, we need something different, and I hope that they will start to think of something different. I think that another very important thing about the US system that you didn't mention is I think of all the countries in the world, it's a country where a non-American feels best. So a foreigner in America feels best in America, whether you're Chinese or anywhere. And that, I think, is an enormous advantage. America is a very immigrant-friendly country. One of the reasons is they don't expect very much. I mean, if you become an immigrant in France and you refuse to speak French, it would be very difficult. In America, no one cares whether you speak it, whether you don't speak it, where you come from, and that is a huge strength.
Thank you. Actually, we should finish our session. Maybe the last question from the audience, please. Anyone?
I'm here.
Okay.
Okay. I'm Michael Spinotti from Brazil. We have the same question of MFTI in Brazil and how to attract and to be more innovative and how to transform science into innovation. And I have two questions to you, Michael. Thank you very much for your inspiring lecture. One, why are you in Stanford? And what should be a cool place to be? What are the characteristics of a good place to be to develop your science and make it happen into innovation? So we could think differently on how to create the conditions of course, critical mass imports, but what's more we can need?
So I think for me, Stanford offered a level of freedom. In other words, you know, I am full-time at Stanford in terms of my e-time, but physically they do not care where I actually am. And that is something which most places would not like, the idea that somebody was full-time there, but was not physically there all the time. I, for this reason, pioneered distance teaching. It's now boring for me, but it was something I did 10 years ago. So I think they're very liberal that way. They're very supportive. They're also a place that is changing all the time. They're very innovative because of the people there. On the other hand, although Stanford is very rich, and to give you an idea, the president of Stanford raised $6.5 billion in donations in 4 years. And he raised them from private people, not from oil or something like this. Is a lot of money. But the fact is that I'm sorry. The fact is that if I went to the president of Stanford and said, I want my own building, he would say, should I call the taxi for you to leave, or will you leave by yourself? And there are examples of people who have Nobel Prizes who wanted additional facilities and they left. So Stanford, although it has all the money is for new initiatives. If I said, I want to create a center for something and I'm prepared to work really hard and hire people, I would get the money. But if I said, I want a building, I'm important, forget about it. And that's very important. So it's a strange mixture of liberalness, openness, but also a certain integrity. And it's important to realize that, you know, I guess in Stanford, in the medical school at Stanford, there have been 5 Nobel Prizes in 7 years. That's a lot, out of a faculty of about 100 people. So that's probably one of the highest rates, perhaps except for Cambridge. They give you a lot of respect. They try to make money out of you, but you don't have any power, which is great. It should be like that.
So, an answer from Robert.
Well, back to the basic question of how should research be financed, by grants or institutional, or should it be a mixture? Now what we see in countries where there is essentially grant-based financiation, that people spend a substantial part of their time, which they actually should devote to research in writing grants, with a success rate in the order of 10 or 20%. So this is not really fruitful. On the other hand, purely institutional financiation is a great danger, because there is little control. I think a mix of this, and I'm happy to say that in Germany we do have that. We have institutional financiation in the research institutions like the Max Planck Society, and we have mostly grant financiation in the universities. So I think this works quite well, I would say.
Okay, some more questions?
For me, MIPT. I would like to return to some kind of scientific questions. So first of all, thank you for great review in the field. And I want to ask you about the possibilities of computational study in biochemistry. You mentioned about simulation of many different systems, but what do you think? Is it possible at the current stage to predict some novel types of biomolecules for using in, I don't know, biotechnology, or maybe they are used in cells, but we cannot determine the structure or whatever? And if so, which kind of biomolecules can be the most promising in this field?
So I think that's again a very interesting question. I will not answer the second part because I don't believe that I know, but I think that one issue that was alluded to a little bit by Professor Huber is that currently we are quite good at predicting to some extent properties of certain classes of molecules like proteins. It's much more difficult for novel molecules, and for example, drug molecules are all very novel. And I think what we need to do, and this is actually something which I did want to describe a little bit, is instead of having a dead physicist's force field, we need a quantum mechanical force field. And the fact that I've been to Moscow so many times came from a company that was actually founded in the United States by Carrie Queen, who was the person who made a lot of money from humanizing antibodies. And then he decided that you would get better chemistry through physics. And he created a group in downtown Moscow, near Tverskaya, of 70 physicists who ended up trying to derive a force field for molecules based on quantum mechanics. Now, it turned out that after a while, Carrie Queen got tired and he invested $10 million and it was a bad time. And he left the company, but it's actually started out again. And they have a small group in Moscow, just 4 people. But I think this kind of new approach to combining physics of, quantum mechanics is physics, it's not chemistry. Combining that to new kinds of atoms is going to be very important. What has happened is that nowadays quantum mechanical methods take a lot of computer time, but they are so good that you can calculate energies of molecular systems much more accurately than you can measure them. So I think when it comes to new systems, we're going to be at a new level of being able to deal with this. It'll also be very much driven for drugs, because a drug molecule needs to bind very strongly, so you can use a very small amount, so you will have no side effects. And very strong binding is going to depend on all sorts of weird physics. It's going to depend on interactions which are special. So I actually think that the next decade is going to involve a whole new kind of chemistry of force fields that will allow one to deal with novel systems. And I think this could be very interesting because people talk about synthetic life, but usually it's just making DNA. It's not really synthetic life. A new class of biomolecules or molecules that are not bio could be amazing. That's a mind-blowing idea.
Thank you very much.
Well, there is a quite fascinating new field emerging, namely the production of non-natural amino acids and making proteins with quite exotic properties. So the techniques are available already, and I think we are already able to generate a whole new universe of biomolecules by using non-natural components.
Okay, thank you.
And the real last question. So who will be the luckiest? Ivan?
It's Ivan, MIPT. Dear Nobel laureates.
Microphone поближе.
Oh yeah. Dear Nobel laureates, is there a feeling that the whole creativity is left only for the organic chemistry, not to, the inorganic or analytical chemistry in the world?
So did you catch the question?
Am I clear? Okay.
Now the chemistry that we have in living creatures is essentially carbon chemistry. And that has a good reason, because carbon is the molecule which is by far most versatile in making bonds and new structures. No other element can do this, silicon, not, for instance. So complex molecules in living systems are carbon-based, and this has to do with the basic principles of chemistry and physics of the elements. I'm not sure whether this is an answer to your question, but I think it's worthwhile to consider.
It was probably if there is any creativity left on inorganic level of the chemistry.
Well, there are fantastic achievements and molecules made in inorganic chemistry for material science, for instance. But by far not of the diversity and versatility that we have with organic carbon-based molecules. So material science, new materials.
Thank you a lot.
Thank you.
Just to add something very simple to that, if you just look at the periodic table, at Medvedev's table, organic elements are the majority of the simple ones. If you look at molecules found in outer space on meteorites, they're amino acids. So I actually think in some ways that, just as Robert said, these are basically the fundamental molecules that can do complex things. You know, I know that there was a lot of work on boron chemistry. You know, maybe, but why? I don't know.
I think the general answer is if you have creativity, you can apply it to everything, even for creating this conference. So, so thank you so much. I just want to add that it's just the beginning of our conference. And tomorrow we'll have a lecture of Robert Huber in the beginning at 10 o'clock. So we'll also could ask him question directly after the session, and to another speakers from University of Wisconsin, from Norway, from Brazil, from MIT and other institutes and universities. So we are very welcome you tomorrow. And now we have a coffee break for 15 minutes. So the next session will begin at 12:30. So this session will be dedicated to pharmaceutical clusters and the centers of excellence of another universities in Russia that are supported by the Federal Program Pharma 2020. And I would like to thank all our Nobel Prize winners and other speakers for participating in this morning panel, and everybody. So we are waiting for you in 15 minutes. Thank you.
Thank you.